Here you can see a microfluidic droplet generation and monitoring setup. This system consists of a pressure-based pump unit, a microfluidic chip device and a compact optical system. The fluids are guided via P-capillaries into the microfluidic chip device. This is a fully functional inverted microscope consisting of continuous white light source and an integrated microfluidic chip device. The chip is mounted on an XY stage to move it laterally. Additionally, there is a possibility to adjust the Z focus precisely. Underneath the stage, a long-distance microscope objective is integrated. An Ethernet port is used as a data transfer interface. The imaging profile M is available in different versions and therefore customizable. The modules are available with 5, 10 and 20x magnification. Furthermore, with different light sources and wavelengths as well as fluorescence light sources are possible. Here you can see the live image of the imaging module. The droplet generation takes place on a microfluidic functional structure, in this case oil and water phase. The droplets therefore are formed at the micro nozzle. In addition, there is a second dosing unit here which enables the direct dosing of active substances into the existing droplets. For analyzing the droplets, we have a special plugin for our OptiViewer. This software plugin can be used for the live classification of the droplets. By activating the individual functions, they can be displayed in the live image. For example, the bounding boxes can be displayed. Also, each droplet is automatically assigned to a sequential ID. This is important for the process monitoring and the later evaluation of the experiment. In addition, the area and therefore the volume of the droplets can be data mined, for example, to monitor the stability of the whole process. And of course, all measured values are summarized in a table. This table can then export it as a CVS file for further processing.